Welcome to Spring LIB.
Hello students, the given question talks about blood group. So let us answer the questions one by one. So the answer to question 1 is the possible genotype for the blood group A positive will be homozygous for A, RR, or it can be A, O since it is recessive, and then heterozygous for the RH. And the next for O positive it will be OORR or OORR, heterozygous for RH. The next B negative, this can be B recessive for RH, or it can be BO recessive for RH. Next AB negative, this can be AB recessive. Now coming to the fifth question, wherein we have a parent with A positive blood group and another parent which is O positive. So their offspring will have what blood type? Before we come to that, let us first write down their genotype. So genotype of this parent, say let's say is AARR and this OORR. Then the offspring will be, this is the first type, type A positive. And we can have another case. The second case is when the parents have the genotype AORR and OORR. Then the offspring can be AORR and then AORR and OORR and OO. This one will be A positive, this will be A negative, this will be O positive, and this will be O negative. Coming to the next question. A test cross is a cross to know the genotype of a dominant phenotype, because this dominant phenotype can either be homozygous or it can either be heterozygous. So in order to know whether it is homozygous dominant or heterozygous dominant, a test cross is performed between a dominant trait and a recessive trait. Let us take for example, if we cross a recessive dwarf plant with a heterozygous tall plant. So the offspring will be heterozygous tall and dwarf. So half will be tall and half will be dwarf. And if we cross a recessive dwarf with a homozygous dominant, then all the offspring will be tall. Here we are getting 100% tall. Here it is 50% tall and 50% dwarf. So this is how a test cross is performed in order to know the genotype of the dominant phenotype. So in this case, this is unknown. This is unknown, but if all the offspring are expressing the dominant trait, then this means that it is homozygous. And in this case, this is also unknown. The genotype of this is also unknown. So if we cross it with a recessive trait, if half of the offspring expresses dominant and half of the offspring expresses dwarf, then this shows that the dominant is heterozygous. Sex chromosomes are chromosomes which determine the gender of a person. These are chromosomes which determine the gender. Say for example, males have XY sex chromosome and females have XX sex chromosome, which identifies this as a female and this as a male. Whereas autosomes, these are chromosomes other than the sex chromosomes. Say for example, humans have 46 chromosomes, or we can say they have 23 pairs. So chromosome 1 to 22, these are autosomes. And chromosome 23, these are the sex chromosomes, which determines the gender of a person. Now coming to the next question, the difference between metaphase of mitosis and metaphase 1 of meiosis is that in metaphase there is just one pair of chromatids and there is no crossing over, whereas in metaphase 1 there are homologous pairs of chromosomes, thus there occurs crossing over. Now coming to the difference between anaphase of mitosis and anaphase 1 of meiosis is that in anaphase there is a separation of sister chromatids, whereas in anaphase 1 of meiosis there is separation of homologous chromosomes, while the chromatids or the sister chromatids are attached. Hope you found the video useful. Thank you.